So today I will discuss about your mitochondrial DNA and chloroplast DNA that is given in your genetics syllabus. This video is for the third semester students of biotechnology. So let's proceed. So about the mitochondrial and chloroplast DNA, you all know that according to the endosymbiotic origin, this mitochondria and chloroplast have come into our mammalian into our normal cell. Okay, they were once upon a time they were actually they were some other organisms. So origin of mitochondria from alpha proteobacterium. So it has been observed that mitochondria has been derived from an organism whose genome is similar to your alpha proteobacterium, whose genomes are almost same. Though through endosymbiosis, this alpha proteobacterium converts into mitochondria within the eukaryotic cell over time. Just like that, chloroplast also come into existence into plants. Let us discuss about mitochondria first and then chloroplast. So mitochondria contain their own genetic system. It is totally distinct, totally different from the nuclear genome of the cell, and the DNA which is found within the mitochondria are called mtDNA or mitochondrial DNA. Most of the mitochondrial DNA are circular in nature, like those of bacteria. From this we get a provement that the bacteria also have their circular DNA that is present in multiple copies per organelle. So within the mitochondria, multiple copies of circular DNA found within the mitochondria. Size of this mitochondrial DNA varies from species to species. It's not all the species will have the same amount of mitochondrial DNA. It varies. Like for instance, the genome of human and most other animal mitochondria have only 16 kilobases, whereas larger mitochondrial genome are found in yeast, approximately 80 kb, and in plants that is more than 200 kb. So it's not like that higher organisms will have great amount of mitochondrial DNA, low order organisms will have small amount of mitochondrial DNA. That's not the thing. In yeast, they have a large mitochondrial genome, about 80 kb, whereas in plants also they have more than 200 kb of mitochondrial DNA, whereas mammals like human and other animal, they have mitochondrial DNA as about 16 kb only. Well, large mitochondrial DNA does not mean they will code all of them the proteins. Most of the mitochondrial DNA are non-coding region. It should be noted that larger mitochondrial genomes does not code for more genes or protein. Okay, it should be noted. Like for instance, in Arabidopsis thaliana plant, about 31 proteins are encoded by the mitochondrial DNA gene. In human mitochondrial DNA, about 13 proteins are coded by them. Okay, so it's mean to say that mitochondrial DNA does not code all of the proteins. Okay, some of the things needed from the cellular DNA as well. Now, the large mitochondrial genomes are composed of non-coding sequences which form the introns, which forms the spacer DNA or linker DNA, and does not appear to contain significantly more genetic information. In earlier days, well, you have learned about the C value paradox. I have discussed with you. Uh, in early days, scientists used to think that large DNA means more information they will carry, but that does not mean larger DNA may contain non-coding sequences, which will not code for any protein. Okay, so always remember that large mitochondrial genome, most of them composed of non-coding sequences, which appear in the form of intron, which appear in the form of spacer DNA or linker DNA, and does not appear to contain that much of genetic information. Well, a table has been given here. According to this table, you can see the size of the mitochondrial genome on your right side. On the left side, the scientific name of the organisms have been given. Like Arabidopsis thaliana have about 370 kilobase pair of mitochondrial DNA. In yeast, Saccharomyces cerevisiae, about 85.7 kilobase pair of mitochondrial genome observed. Higher organisms, Homo sapiens, it contain about 16.5 kb kilobase pair of mitochondrial genome. Fruit fly, Drosophila melanogaster, it has about 19.5 kilobase pair of mitochondrial DNA. Then from the Cucumis melo, it contains so much high amount of mitochondrial DNA, but most of them are not coding. Now, about the codons, as you know, codons are the three nucleotide long decoding elements that specify particular amino acids. These are also found in mitochondria, but it does not follow the same universal codon system that we normally know. Okay, like for instance, UGA. Well, you all know that UGA, UAA, and UAG codes for stop codon. Universally, it codes for stop codon. But in case of mitochondria, particularly in human mitochondria, UGA code for tryptophan. So it is not following the universal codon system as we know. AGA, normally AGA codes for arginine. Universally, it is accepted, but within the mitochondria, it codes for something else. In human mitochondrial genome, AGA codes for stop codon. AGG, AGG also codes for stop codon by the mitochondria in human. So we can see about 4 stop codons are observed in human mitochondrial genome. That is UAA, UAG, and the rest of the two that we can see here, AGA and AGG. AUA, which codes for isoleucine normally amino acid, here in mitochondrial genome of human, it codes for methionine, the start codon. So, just as I have said, they are not following the same universal codon system as we have seen in nuclear DNA. Now, let us discuss more about the human mitochondrial genome. Human mitochondrial genome encodes about 13 proteins, and these 13 proteins are involved in the electron transport chain and oxidative phosphorylation. Well, we have studied about electron transport chain and oxidative phosphorylation in our higher school, okay, and how it carries, how it breaks to get that ATP. So out of the different proteins involved, 13 proteins that are needed for electron transport chain and oxidative phosphorylation, these 13 proteins are encoded by the mitochondrial genome. The genes that encode these 13 proteins are transcribed and translated within mitochondria because they have their own ribosomes and tRNA. So ribosomes have their own tRNA, they have their own ribosomes. So mitochondria have their own ribosomes as well as they have their own tRNA. So that's a point to be noted, that unlike other organelles found in the cell, mitochondria have their own DNA as well as they have their own ribosome and tRNA as well. So that's indicate that according to the evolution, mitochondria must have come from some bacteria, originated from bacteria. Human mitochondrial DNA also encode the 16S and 12S rRNA and all the other tRNA which are required for translation are encoded by the mitochondrial genome. So if you see the human mitochondrial DNA, it encodes all the tRNA. It also encodes the rRNA, which is your 16S and 12S rRNA that are required for the translation process. Now, plant mitochondrial gene expression is dependent on nuclear genome. Most of the tRNA that are required during translation of mitochondrial genome are imported from the nucleus through cytoplasm. Now, in case of plants, it's a bit different. In plants, their mitochondria does not make enough tRNA. Most of the tRNA that are needed for translation of the mitochondrial genome data that are imported from nucleus to the mitochondria. Okay. Plant mitochondrial genome encodes 16S, 12S, and 5S rRNA, and the proteins which are required for transcription translation, like the RNA polymerase, then ribosomal proteins, translational factors, are all encoded by the nuclear genes that are further expressed and transported into the mitochondrial matrix. So in case of plant mitochondrial DNA, it is a bit different. Unlike animal cell, plant mitochondrial genome encodes only that 16S, 12S, and 5S rRNA. The other proteins which are needed for transcription, translation, like RNA polymerase for mitochondrial transcription, ribosomal proteins, translation factors, these are all encoded by the nuclear genes in the plant cell and that are further expressed and then they are transported into the mitochondrial matrix. So this was about the mitochondrial genome. More information of mitochondrial genome is yet to be said now. So about the mammalian mitochondrial genome. Mammalian mitochondrial genome is a double-stranded supercoiled circular molecule and has a length of about 16.6 kilobase pairs. Normally, the mammalian mitochondrial genome composed of heavy chain or heavy strand and light strand. And why we name them as heavy strand and light strand? Based on the proportion of high molecular weight nucleotides. The weight of nucleotides depend on the strand. That's why we name them as heavy strand and light strand. Okay. GC repeats more weight, molecular weight. Okay. So about the heavy strand and light strand found within the mitochondrial genome and of mammals, and it is of a supercoiled double-stranded circular DNA of 16.6 kilobase pairs. The heavy strand found within the human mitochondrial genome has the gene coded for coding for two rRNA sequence, 14 of the 22 mitochondrial tRNA, and all the protein encoding genes with the exception of ND6. So it is saying that the heavy strand contains the heavy strand found within the mitochondria contain the genes for encoding two rRNA sequence. Then mitochondrial tRNA, most of the mitochondrial tRNA are encoded by the mammalian mitochondrial DNA. 14 out of the 22 mitochondrial tRNA are encoding by the mitochondrial gene, and all the protein encoding genes with the exception of ND6. What does ND6 encode? That I also don't know. I have to see. I have forgotten actually. Uh, it's sorry to say, but I will discuss with you. Okay. There is also a large non-coding region found within the mitochondria of about 1 kb that contains the regulatory elements for initiation and termination of transcription of both the strands. So there is also a non-coding region found within the heavy strand about 1 kb that contain the regulatory elements for initiation and termination of transcription of both the strands. So we have to see about the ND6. What does ND6 encode? Well, if you know, let me remind. Then now the D loop or the displacement loop or the control region is within this non-coding region, and it contains the origin of ODH or heavy strand origin of replication, where the replication process is initiated. So within this non-coding strand of chain heavy strand, don't say heavy chain, of heavy strand, within this non-coding region found within the heavy strand of mitochondrial DNA, there is a D loop. D loop means displacement loop, uh, that contains the origin of replication for the heavy strand, where the replication process is initiated. There is also another origin of replication for the light strand called which is the second origin of replication and known as the ORI-L. So two origin of replication, quite surprisingly we can see origin of replication for the heavy strand and origin of replication for the light strand, and this is found within the non-coding regions. Okay, ultimately they will not code for any protein, but they are required for the replication initiation process, right? Now just like we have nucleosomes in the chromosome in the nucleus, mitochondrial DNA genome is packaged into nucleoids. Well, nucleoids can be compared with nucleosomes. Nucleoids mean mitochondrial DNA with somewhat histone like proteins to form nucleoid like structures to pack the DNA within the mitochondria, right? So, mitochondrial DNA genome is packaged into nucleoids which are dynamic in structure, which are normally formed by the mitochondrial DNA genome, and the proteins which are involved in replication and transcription. So, most of the protein which are entangled with the mitochondrial DNA to form nucleoid, most of the proteins are related with replication process, transcription process of the mitochondrial DNA. Although these structure provide a level of organization, it is not as tightly packed or organized as nuclear DNA. It, if you compare with the nucleosome, the solenoid structure which we have seen in our chromosomal DNA organization, it's not like that of organized structure. Here and there they are not so much tightly packed. Here the nucleoids where the mitochondrial DNA entangled with the protein to form a compact structure, not like that of tightly packed or organized manner they are seem to be seen compared to nuclear DNA. The nucleoid protein, that means the protein which involves to form the nucleoid by interacting with the mitochondrial DNA. So this nucleoid protein include your polymerase gamma. Polymerase gamma is the main polymerase for mitochondrial DNA replication. The single-stranded DNA binding protein, the single-stranded DNA binding protein which is found within the mitochondria. They also bind with the mitochondrial DNA to form that nucleoid structure. Twinkle, well, this name I get first time I get this name. Twinkle, which is actually a DNA helicase, okay, that we know. Guidase is also a type of DNA helicase. Here we name them as Twinkle, PHB, prohibitin, and TFAM. This is another protein which normally found to be organized with the mitochondrial DNA to form nucleoid. TFAM full form transcription factor that regulates the mitochondrial DNA expression during transcription process. So here we can see that about the nucleoid structure which can be compared to the nucleosomes, but they are not tightly packed. And which are the proteins which form the nucleoid structure? The proteins which are involved in replication, which are involved in transcription, like your polymerase gamma, involved in replication, single-stranded DNA binding protein, DNA helicase, TFAM, which is the transcription factors which are needed for regulation of the mitochondrial DNA expression. Now, the structure and organization of the mitochondrial DNA nucleoid is an emerging area of research with new proteins frequently being identified. Nowadays, we are identifying the structure and organization of the mitochondrial DNA. It is an emerging area of research. Many things come into play, come into existence when we are studying about the mitochondrial DNA. Well, one new thing I want to say, I think you know, but well, when we get ill, we used to consume fever tablets, okay, like paracetamol or aspirin, okay. But these tablets can hamper our mitochondria. That's why we feel weak, especially when we consume antibiotics. Also, it also uh, makes us weaker because of disruption of the mitochondria organelle. So somehow mitochondria is related. We all know mitochondria is related with the powerhouse. It is related with the energy production, ATP production. But at the same time, if you see in the molecular level, some of the genes encoded by the mitochondria, which are nowadays, it's being we are seeing that they are performing some important functions. So the nucleoids are located on the matrix on the interior side of the inner mitochondrial membrane in close proximity to the oxidative phosphorylation system. So most of the nucleoids that we can see here, they are located close to the matrix on the inner side of the mitochondrial membrane, where your oxidative phosphorylation system is located actually.